Dr. Barry Byrne. He's a clinical scientist studying a variety of rare diseases with the specific goal of developing therapies for inherited muscle diseases. He's a pediatric cardiologist, and his focus is on conditions that lead to skeletal muscle weakness and abnormalities in heart and respiratory function. His group has made significant contributions to the understanding and treatment of Pompe disease, which is a type of neuromuscular disease. The research team has also developed therapies using adeno-associated virus-mediated gene therapy to restore cardiac, that's the heart, and skeletal muscle function in muscular dystrophy, Friedreich's ataxia, Pompe's disease, and other inherited neuromuscular diseases. His group at the Powell Center, Powell Gene Therapy Center at UF, has also established a series of new methods for large-scale development of this virus, AAV. And his work is supported by several NIH grants and from the industry and foundation. Dr. Byrne is currently, Dr. Barry Byrne, B-Y-R-N-E, is the Associate Chair of Pediatrics and Director of the university's Powell Center and Child Health Research Institute. He obtained his BS from Denison University and MD and PhD from the University of Illinois. Then he completed a pediatrics residency from Johns Hopkins and also did postdoctoral training in biochemistry there. Following his early career at Hopkins, Dr. Byrne moved to the University of Florida, and currently he's the Earl and Christy Powell University Chair in Genetics. In addition to his academic appointments, he serves as the Chief Medical Advisor of the Muscular Dystrophy Association and a member of the TACT Committee of Treat DMD, which he'll tell you what that stands for. Barry, please welcome Barry Byrne.
Thanks so very much, Pushpa, and thank you all for joining that are here in person and also online. As Pushpa said, I have a few responsibilities at the university, and I'm happy to tell you about how Ken Burns had a vision for this area of medicine, which I think when we all started in this in the late 90s, we only imagined that we could change some of the problems that are faced by children with rare diseases. So that's why I've subtitled this Ken's Vision. And if this doesn't work, I may have to go to the podium. Yes? Stranger's not working, but give it a second. Yes, it is. I'll turn it on again. There we go. So this is a work in progress. I was talking to Dr. Srivastava about how this site called Academic Family Tree helps us build the heritage that any individual makes in their trainees. So I confirmed with Laura that Charles Thomas was one of Ken's mentors, and then there are many secondary connections to Ken that I emphasized a little at his memorial and as part of an article we wrote when he passed away that really called attention to the fact that his leadership helped establish not only this center, but many other clinician scientists and basic researchers around the country. And I think if we really fill this family tree out, we'll see in the cousins and the second cousins of that were directly related to him in his lab, we would be able to, the list would go on. And this has had a huge impact in this field of molecular medicine that I'll tell you about. This young man actually lives in the area. He was the first patient with Duchenne who participated in a study of genetic medicine being developed by Sarepta, the very first treatment for Duchenne. This had some effect, some benefit, but it's not a transformative effect, and I'll explain to you what the difference is. when we consider the magnitude of effect of a genetic medicine that can directly affect a gene that causes a disease. But this is really what I wanted you to have this background about how this field was born here at the University of Florida, where we are today in treating patients with inherited diseases, and what the future holds. So if we think of the past, it was really right here in the academic research building, and this was before the cancer genetics building was built or before the Brain Institute was built, which is in the foreground. And way over there on the right is where Arun's lab is. And those two places actually didn't exist when we first started this initiative. We were all based here in the academic research building in the very center. That was pretty much as a result of Ken's vision of how to build the basic science departments that the cancer genetics initiative was started, in which he ultimately became the first director of that undertaking, along with the interim role that Nick had. Nick Maziska, one of his trainees. So this complex developed, around the conventional disciplines, really started to expand into this area of how could we, across the various disciplines. I think you heard recently about the opportunities in ophthalmology, but now we have considered programs both for neurological, musculoskeletal, rheumatologic, and cardiovascular diseases that are part of this initiative. And you may not have known, being in Gainesville, that you're really being a really true blue gator. Your DNA is also orange and blue. And now we know how to manipulate DNA in ways that we didn't really understand 20 years ago. And both for the recessive diseases where both parents might be carriers, we also now have ways of treating dominant conditions. And this is really well exemplified in how this, particularly the most severe forms of inherited diseases, affect children. So we know that there are about 400 million people with a rare disease, and half of them are children. One of our biggest populations of patients with inherited neuromuscular diseases, so that causes generalized weakness, is Duchenne muscular dystrophy. And every day in the US, we lose a patient with Duchenne because of the lack of effective treatments. And like that boy I first showed you, we can do better than the first round, the first generation therapies. When, as a result of the building of the Health Science Center, Dr. Nelson really focused on the UF Health's research mission. And these 6 pillars were identified as areas where we wanted to exceed. We know about the excellence in the neuromedicine initiative and precision health related to pharmacogenomics. Now in the data sciences area, the culmination of this initiative with Malachowski Hall, and if any of you have seen that on campus, it's quite unusual and amazing home for all the data sciences. The cancer genetics building came online, and now we have a comprehensive cancer center. There's always been a very strong area of expertise in diabetes. And one of the pillars was to advance the knowledge and ability to use gene therapy in clinical populations. And as Pushpa said, that my background actually is in a clinical area that was not traditional for this kind of work, because many of the causes of both functional and structural heart disease were not known. But the heart is a muscle too, like the rest of the musculature, and there's a lot of overlap between the conditions that cause generalized weakness and heart muscle dysfunction. But internally, we collaborated across a variety of areas, both the Children's Hospital, the Cardiac and Neuro Hospital, as well as our regional network. And then in the outpatient setting with medical and surgical specialties. But really what we've tried to do is engage also the community to understand how these products or drugs that are being developed might impact the lives of others. We do that particularly in the most vulnerable population through newborn screening. And I'll mention how that's changed the lives of children who have spinal muscular atrophy. Of course, we rely on our pharmaceutical partners and biotechnology partners, and I'll touch on that a little later because of the strong position that UF Innovate has had in helping us work with those industry partners. And in the future, in this kind of cutting edge therapy should be for every condition with a high unmet need, because traditionally we've had no means to treat any aspect of these diseases. And then as an academic health center, we have to mentor and train the next generation of providers and scientists. And a lot of this, in fact, maybe you would say all of it now is based on this tool, which is represented here in an amazing imaging technology that was developed partly because of Ken's vision and his willingness to bring Mavis Sabanji McKenna and Rob McKenna to the University of Florida. And this unique structure of the outside of AV has also enabled us to make even better versions of this through something called directed evolution. This tool enables us to bring initially only small genes into muscle cells and other cells all around the body, but now even larger genes and up to the size of the biggest genes in the body. Particularly the story in Duchenne is shown in this image on the right. These are all the investigators and families who imagined in 2016 that there would be a treatment for Duchenne dystrophy. And the young boy held by Ilan Ganot there and his wife Annie was just 7 at the time. And we know that each of those parents and scientists there thought we could move rather quickly into treating Duchenne, and the complexity of it is quite high. But this year, we now have a gene therapy treatment for Duchenne, as well as the one that helped that young man from our area. And then this got some popular attention in a movie called Extraordinary Measures about a family that was affected by 2 children with Pompe disease. And so this, if you have a chance to watch, see if you can find me in the beginning of the movie. I really was a pleasure to participate in that story as an amalgamation of all the scientific activities. Harrison Ford played the kind of grumpy scientist. This is a recent analysis that we've never been that good about marketing our own accomplishments, but this paper from last year provided a network analysis of where did all the knowledge come from in the AAV space, and everything that's in red here and all of the affiliated partners of the University of Florida is the biggest cloud of knowledge came from all these groups. And then the second biggest is one of Nick's graduate students, Jude Samolsky at UNC, and then some activities at Penn. But we have the most notable of any institution in terms of number of publications regarding AAV. And I'm happy that at least our group is also one of the most cited. And that's really a measure of the impact. So this was very nice to see this story from 1991 to 2022. And just to touch on some of those big milestones. So when I came in 1997, and Arun, you said 99? 2002, right? Was when you? No, not the first time, the second time. Yeah, Arun arrived here twice. But Jennifer Hunt, who's the Dean of the College of Medicine, commented that at the time we started these programs, we adjusted what we were doing based on what was happening at that time. And there was a death of a patient at University of Pennsylvania. This would have halted really what we were doing unless we were imagining what the future would be, not where we are today. And the great Wayne Gretzky said, you know, always skate to where the puck's going, not to where it is. And I think we had this vision because of Ken's leadership of what we could do to improve on this field. And not, in fact, this was pretty much the end of that use of that vector system in people, and the focus became on AAV, which was our contribution to this field in general. And you can see these really important banner years. The gene therapy for inherited blindness approval in 2017. And Dr. Boy must have talked a bit about the experience for LCA. So I was the investigator of the study for LCA, even though I'm not an ophthalmologist, because we had an expert from University of Pennsylvania, Sam Jacobson, as really with Bill Hausworth had conceived of this therapy, and Bill was one of Ken's trainees. And so that was the very first approval. Then this was a watershed moment. In 2019 was when the first systemic gene therapy was approved. This version of gene therapy was not given locally, but given to every cell in the body that could be exposed through the blood system. So this is an intravenous delivery of a gene therapy agent. Some aspects of this milestone were accomplished here in some of our scientists who went to Nationwide Children's Hospital, where this work was done, helped facilitate the early studies that you'll see some examples of how transformative they were. And then what can we do in the future? I mentioned now we have one approval for Duchenne. We need to be able to improve access to drugs like this, because in other developing economies, the high cost of these products can't be afforded. I'll show you an example of one of the ones we worked on here, a form of congenital Parkinson's disease, and this treatment costs $4.5 million. Obviously, the life of a child, we can't put a value on, but those expenses, I think, can't be borne by some places where even the simplest forms of healthcare aren't available. So we need to do more to lower costs, and that's something we're very actively working on. And you can see the pace of discovery is increasing. So Luxturna, the first one with an AAV vector in 2017, and then this treatment for SMA in 2019. And once this got rolling, you can see 4 or 5 approvals in every year. And I think we'll see that this pace continue to increase because we're building on the knowledge from the prior authorized products. And these are ones we had a role in. I mentioned LCA and the efforts then put forward by Spark. We developed a product for a form of X-linked myopathy called X-linked MTM1. This AADC product was just approved for this congenital Parkinson's disease. And then, as I mentioned, the SMA therapies, as well as 2 products in Duchenne. So this is what the first approval looks like. Luxturna is given in a part of the eye under the retina, a somewhat protected space where the gene replaces a missing gene product that is involved in recycling of the vision receptors. And this was really quite striking that it restored vision in some patients who had extensive vision loss because of this retinal pigmented epithelial cells dysfunction. And then we did both the first commercial infusion of Zolgensma, and this baby from the area was just diagnosed weeks before the authorization. And we had anticipated having the infrastructure to get authorization. Zolgensma is just $2.1 million. And that, of course, required a lot of work with the health system to figure out how to acquire the product, pay for it. And now I'll show you going forward what we're able to do in this regard. And the biggest population we'll have in Florida, about 1 new patient per week, has this form of muscular dystrophy called Duchenne dystrophy and Becker muscular dystrophy. This is the product developed by Sarepta. Last year, we were able to use it in 4 and 5-year-olds, and there's a limited population that are that young. And this year, it's been expanded to all ages. And in order to even keep up with the new patients that are being identified, we've worked out a means to treat up to 1 patient per week, which is a huge undertaking by the team. And I'll show you the team that it takes to do that. And here's what's anticipated in the future, particularly this product that was approved in Europe, Upstaza, is something we started here for patients with congenital aromatic amino acid decarboxylase deficiency. This is one of those children. Normally, these children would not be able to move at all. And they do breathe on their own, but they can't move. And in SMA, not only do these children not move, but they require a ventilator to live. This couple were from 2 different parts of the world, but yet they each carried the gene for AADC deficiency and had this girl who was affected. And our collaborator in this is Paul Hu. He's a geneticist who I knew from Hopkins, who's based in Taiwan, where many of these patients have been identified. Mom is actually Thai, and the dad grew up in Osaka, Japan, even though he's Caucasian. And this girl was so delightful at this ceremony 2 weeks ago in Taipei, just behaving and acting normally. An amazing part of this story is that it's the first product that's been approved with a device that's needed to deliver it, because the delivery is done by a neurosurgeon in the very deepest part of the brain called the putamen. So just that 1 dose of the vector given in the center of the brain is restored movement in this girl. So, well, how's the team built? So it took a lot of teamwork to do this, as I tried to emphasize in the beginning. We have the preclinical group at the Gene Therapy Center. They're the laboratory scientists who are at the bench doing studies in mice and cells and understanding the mechanism. And then that group works together with the clinical team that is doing either clinical care in the medical plaza that you all may have seen, and then this building where we do on this side clinical research in the NIH-funded GCRC, and all of our industry-sponsored studies are done over here. And I'll tell you about an exciting new development with the hospital that, as commercial gene therapy, is now being done on this side of the building, which was formerly the Institute on Aging. So here's the preclinical research team. You can see it takes a lot of experience and expertise. We have early career scientists, more experienced individuals who are all part of this. And one of the students here, Chandra Trantham, for example, herself has a rare disease. So she's studying a rare disease, but also has Friedreich's ataxia, which limits her mobility. But she'll be graduating this fall. The clinical research team sees patients who are involved in research studies. Some of the same people do both, like myself, but this group is run by Julie Berthy and a series of research coordinators, and we even have a comfort specialist who's Bailey Coker, who's Mackenzie's dog, who actually is an emotional support dog who volunteers in the hospital. And so a whole variety of specialists, nurse practitioners, speech and language pathologists, and the investigators shown here of various specialties, particularly in neurology. And our collaborations are more than within the institution. They're with many external organizations, particularly parts of the NIH. Some of the significant support we have is from the Muscular Dystrophy Association and many other patient foundations. This requires a lot of money, and we rely on industry partners to help fund these studies. So here's a sample of all of the industry partners that we're currently working with, as well as our academic partners at other institutions. And really throughout the country and internationally is it takes a village, as they say. And then our clinical team sees patients that are our continuity patients from around the region. We have care coordinators, support team that does both imaging and scheduling. Myself and Dr. Shi do cardiology, adult and child neurology, pulmonary, endocrine, nutrition. Coy Holderman is an oncologist. Larry Schumacher, a nephrologist, rehabilitation specialist, physical therapy, and occupational therapy. So there's a whole team of multidisciplinary specialists that care for patients with neuromuscular diseases. And over the past 5 years now, we've accumulated a fair bit of experience, probably more than any other academic health center in terms of the first experimental therapies. We had over 50 patients enrolled in studies. Each one of these is an enormous project and undertaking. We may see a patient as part of a study 20 times, almost every other week in the first year after treatment. Here's an example of those patients which kind of became family members. Each of their enrollment order in a study, they had their photos taken in the stadium on the row where their patient number was. These first patients were in the low dose group, and these last 4 were in the more high dose group. This boy here was the first patient from the United Kingdom to receive gene therapy. This young man, number 4, is actually a junior in high school now, and he went to his prom for the first time this year, when normally at that age, he would be using a wheelchair. So the treatment really changed the trajectory in his life. And as I mentioned, Duchenne is one of our more challenging problems, but we have now 6 different sponsors working in this area. And recently, with one of our former colleagues who's now at Indiana, have the potential to add a new approach to this that will be even more effective. Because of the access to commercial therapy and the cost, we partnered with the hospital to form the UF Health Center for Advanced Therapeutics. And luckily, there was a space that needed to be renovated here on the ground floor of this building. If you drive by that, it's right next to the band practice field. So when we're on the upper floors, you can watch the band practice, and it's a bit noisy inside when the band is out there. But this place gives us the opportunity to really bring cell and gene therapy for our clinical patients to a single location where everyone has the level of training necessary to get the best outcomes. And where do those patients come from? They come from 3 different continents and 20 different states. So not only just the Southeast region, where it's obviously a little more convenient to get here, but we have referrals really from essentially Atlanta down to Miami, coming right here to Central Florida. And in some cases, these therapies are just not at all available in the country where that person might have been born. And we always feel like that geography shouldn't be limiting for their access. So particularly the people from South America and Brazil often have relatives in Florida, and they've come here to participate in studies and often stay permanently. I'm going to make an analogy so you can think about the impact of this in the field of medicine and what a transformative aspect it has. A lot like transplantation, gene therapy treatment is the only medical treatment which is entirely irreversible. Once the infusion's done, those AAV vectors that travel into cells and bring a curative gene with them never leave those cells, particularly in the brain and some of the muscle cells like the heart. And so we really manage these patients a lot like a transplant recipient with the whole transplant team. This is one of the local children with spinal muscular atrophy. And you, also a Gator fan, of course, ignore the orange. That's nothing to do with Tennessee. Just that was the outfit for the day. Um, maybe because it matches its hair. But normally a baby with SMA would never sit independently, it would never walk. And you can see now, 3 years after the initial treatment, um, this child you'd never be able to tell had a fatal inherited disease. How about Duchenne, where there are many more patients? There are about 10 times the number of patients with Duchenne than SMA. And in this region, we have about 450 families just that we follow over time. And since there are 50 new patients per year, we think this Southeast region will have about 43 million people represented and or 900 families. So as we expand, even though we're one of the most populous states, we start to see patients from this bigger region. And then Ohio has just begun newborn screening for this disorder. And we expect that Florida will decide in the next year whether to begin newborn screening. And then it'll become even more complicated because we'll be identifying children pre-symptomatically. Part of the way we assess those patients is another part of an international collaboration with members of the physiology and physical therapy department have developed advanced strategies using a unique technology here at UF and the magnetic resonance imaging lab. This consortium is called Imaging Neuromuscular Disease, and we basically have now expanded this to several hundred boys with Duchenne and studying the natural history of both the heart function, the respiratory function, what happens to their musculature. And what's shown here is this greenish color is the signal that comes when there's fat, and fat normally only is under the skin. But you can see as these boys age, fat and fibrous tissue slowly enters the musculature and inhibits their ability to walk. This also affects bone health. Here's a healthy bone. Here's the thinner bone of a boy with Duchenne, and they're very much at risk for a fracture, excuse me, because of that. And then we can't ignore that our licensing team at UF Innovate has tracked the economic impact developing precision medicines like gene therapy. So in the last calendar year, this is actions of UF Innovate, almost 1,000 team members supporting a number of companies that are either here in the Innovation Hub area, right where Alachua General Hospital used to be, or out in Alachua itself. And you can see that the clients of UF Innovate are really making economic impact of these patents that we all endeavor to describe new things and disclose them to the tech licensing group. But look at the economic impact of the funding for the region from this activity. And some of these are first-time entrepreneurs and certainly companies that contribute to the economic health of the region. Here's another way to look at it. If you look at from the inception of this program in 1995 to 2022, this has had about a $25 billion impact on the region and the university's returns from these investments. So this is an area that we continue to use to justify further investment in this area. So what's next? Now that we have all this infrastructure and these new ideas, we are very lucky that this month, one of the preeminent groups in mouse genetics called Jackson Laboratories, which was based principally in Bar Harbor, is actually moving to Gainesville. Part of their programs will be in Alachua. And we think that a partnership with Jackson Labs will be really important to continue to develop new products based on AAV vectors. And we knew how to add back genes originally, but now with the advent of gene editing technologies, these are all delivered into cells by AAV. We can use something called CRISPR. We can take genes that are overactive or expressing harmful gene products called antisense oligonucleotides. Other forms of RNA can be made from AAV, and this is the next generation. We also know that in some cases, the effect of a gene therapy is not lifelong. In SMA, the target is in neurons within the brain and spinal cord. But in the musculature, as we grow, muscle size increases and the effect is lessened over time. And one of our strong supporters in this area is a family that has a son who was the first to participate in a Pfizer study for Duchenne muscular dystrophy. And they're collaborating with us now to find a way to re-administer AAV vectors, which was one of the principal limitations because of the immune response. Like any vaccination, it prevents the further infection with a virus. And so we've come up with a way to be able to administer the AAV vectors at subsequent time. And so they shared this little video about their son and about the need for developing this technology because of the large number of boys with Duchenne. So this is what they had to say about Connor.
We're going to have hundreds of kids that have received an FDA-approved gene therapy that are also going to need to eventually be redosed.
So the current big goals for KOMD is funding research that will figure out the redosing strategy.
Potentially, we have the opportunity here, if they can figure out the redosing aspect, it could truly change Connor's life and every other kid with Duchenne.
We only have today. We only have today, right? So I try to live in the moment. But I believe we're making an impact. And I know that our organization is going to save lives. And I just hope Connor's a recipient. But I don't know.
So you can see when he was first treated 6 years ago, he could run upstairs. And now he's having trouble getting up on his own. So this is what we hope the future will be able to find the most effective amount of AAV and make sure that it's sustained over the lifespan. So just to finish up, I hope I've been able to tell you that AAV is that we'd like to think of it as a signature program. It's something that's uniquely originated at UF. And that the work we do in the research realm actually helps us discover new things that will become licensed products and commercially available to everyone in the US and around the world. And then there are major challenges that still exist, like this redosing problem and immune responses to AAV vectors that we hope to solve. The young man who you saw in the beginning, you can see his dad there helping him hold up his arm. He now, even though he received the very first Duchenne product, is now a candidate for the gene therapy product. And so we look forward to seeing that if that can help him, particularly his heart function, not succumb to Duchenne as many do. So I'll end there just to show you some of the other kids with SMA who have been treated at the center, all standing and thriving, and without this kind of discovery and miracle in effect to treat a otherwise fatal disease, it's really been very rewarding. So I'll end there and happy to take any questions. Yes. Technical. Maybe we'll share the mic, yes, because the people online can hear. Got it? There we go.
Is any of this going to branch out in the treatment of childhood type 1 diabetes?
That's a great question. So there is a group in California named Korea Therapeutics, and they have developed a strategy for type 1 diabetes. It has going to require some aspect of our input because uniquely they're using two vectors to treat type 1 diabetes. And we've developed a manufacturing technology that would make that possible for the millions of people with diabetes. So there's a strategy that exists, but implementing it is going to be the challenging part. And also that's another area when you have only a handful of patients with the problem, we can kind of spread out the cost. But when there are millions, then we need to lower the cost in order to not hamstring the insurance industry that supports this. So yes, stay tuned. There will be a treatment for type 1 diabetes.
Thank you.
It's wonderful.
Right behind you.
Thank you. I was wondering, with some of these disorders, once a child is older, is it reversed or does it just stabilize?
That's another great question.
Yeah.
So obviously that's where prevention is very valuable. So pre-symptomatic treatment in SMA was necessary to see the greatest benefit because some of the effects were permanent. So I think in the circumstances where there's not reversibility, like in the brain and nervous system, we could hope to stop the progression. And in many cases, that will still lead to a very good outcome. But that's where early intervention comes in.
I was guardian ad litem for a child with SMA. She was about 11 or 12, but you know, she was in a wheelchair and was not even able to turn herself over in bed.
Yeah, so that's an example of where both heart muscle cells and brain cells, once they are injured, it's mostly irreversible. That's where the preventative therapy was real important. I wish it was like Phil Donahue. Audience.
Is the prevalence of Duchenne's more common in one sex than the other?
Yes, so about a third of mothers carry one X chromosome that has the abnormality, but they're not affected. Only the boys are. So much like hemophilia. So if you ever followed the history of the hemophilia in the royal family because of relatedness of people and the maternal carriers for both hemophilia and Duchenne.
Yeah, and we will have the speaker next week, I think, who's going to talk on hemophilia. Right, Arun? Hemophilia. Just 1 minute. On Zoom, just 1 second. Somebody is asking, is there anyone working on later diseases such as Parkinson's?
Yes, there are strategies for Parkinson's disease. So one of the most advanced is when Parkinson's disease is associated with an abnormality in a gene for a lysosomal protein called GBA. And that study's been underway for a while now. And then there are more generalized strategies also for Huntington's disease, ALS, and even Alzheimer's disease. So these are delivered more locally or regionally. And now there are ways using, in effect, really relying on Mavis's discovery of the structure of AAV, ways to add a signature to the outside of the AAV so that it's like a zip code that brings it straight to the area in the brain that's affected. Or it would be done with the same kind of surgical technique that the ADC product was developed.
But Parkinson's is not necessarily, not genetic in origin, is it?
So the theories there is that it may be possible to provide neuroprotective factors.
Yeah, okay. There's another question on Zoom. It's, as a society, how do you believe we should decide how to use our resources to finance gene and cell therapy, including government and industry subsidies and increases in health insurance payments?
Yeah, it's one where at least sponsors who have gotten these products approved have done an assessment of the economic impact and the health cost savings that come with definitive treatment. So in SMA, for example, even though the treatment's at $2 million, the annual cost of caring for a child who's not been treated is almost $300,000. So it was estimated that within the first 3 to 4 years of their life, that really will ultimately save society a lot of money. When we get to more prevalent conditions is where it gets more complicated, because then there's a larger number of affected individuals. And I think it's on us who've really developed this field to figure out also how to make these products less expensive. So one of the reasons the cost is so high is not only they're being used in very rare populations, so they try to recover the development costs. Right now, the methodology for manufacturing products is 20 years old, and we need to apply new technology to that area. And Dr. Srivastava was working hard to increase the potency of AAV vectors is another way to, another lever to pull to improve their effectiveness.
I may have missed this in the question she asked, but with the Duchenne's, at first the first dose seems to alleviate the situation and the child is growing normally. Then after a time, they're regressing and not able to. What's the hypothesis about what's going on with either the cells in the body
Sure.
and or the what's given to them?
So what's been learned from the few hundred kids who participated in the gene therapy study so far for Duchenne is that the expression of the partially full-length gene is not uniform. It's in about half of the muscle cells. That has a near-term effect, but in the long term, those ones that were uncorrected continue to cause inflammation and loss of the effect. And that's one of the notions. And then the other aspect is the fact that the AAV vector is working within the cell in a way that doesn't make it permanently part of the cell division. And so as the muscles grow, this effect is ultimately diluted by increasing muscle set mass. That's why in newborn screening, once that's implemented, we'll go from 3 kilo people to 30 kilo people. We would expect the effect to diminish proportionally. Yep.
Is immune function?
But I will add that this is another manifestation of the discovery of the structure of AAV, because variants of AAV that get not into half the cells, but all of them, are now available to us. And the goal is we'll be able to reduce the dose somewhat, but most importantly, we'll be able to get a uniformity of expression that should be much more durable.
Is there an aspect of the immune function that comes into play also in these patients?
Yes, in a major way, in fact, because some conditions where none of this protein is made at all, the vector itself looks like a new protein, a foreign protein.
Yeah.
And the immune cells go about trying to eliminate it. We've come up with ways to protect against that, but it becomes a little manufacturing challenge because it requires a second vector. But we think that that's possible to solve that problem, within the constraints that we have right now financially. It only requires a small amount of this second vector to lead to permanent tolerance.
Do you follow up with young patients who have received treatment? And if so, for how long?
That's another great question. So this is where, the standard of care is frankly changing. Most studies last for about 5 years. And once the study's over, then it falls back on the clinical team to do the long-term follow-up. And essentially it's lifelong. So in other success stories in medicine, take transplant, for example, at some point, the patients become an adult and they have to be transitioned to adult providers. So this is one of the bigger challenges, is how to manage transition to adult care, because many of these patients didn't survive to adulthood previously. So now adult physicians who may never have seen a boy with Duchenne because they normally die by the time in their early 20s. So this has been a challenge to try to educate our adult counterparts into how to care for this unique patient population.
In addition to the immune response to the vector, I know in some of these genetic manipulations, they're inducing a change in a gene to produce a different protein with different
Correct.
epitopes. How will they modify that?
Yeah,
immune
that's where we've
response?
learned, and if you think about when we first introduced foods, they're given one at a time, partly to allow the intestinal system and the liver in particular to process those new exposures. We're tricking, in a sense, the immune system into thinking the protein has been there all along through expressing the protein in liver cells. So luckily, a good substantial part of the vector goes to liver cells, and it's relatively easy to make that work. And I have to say, you guys ask harder questions than the medical student.
Another question on Zoom is the gene therapy a once and done?
Yeah, so that has been the traditional thought for neurological disease, and that probably is true in the brain and in forms of inherited heart disease. In the musculature, in the kidney or the liver, we know that it's not lifelong. So it's been hard to undo this notion, because when we were first trying to convince pharma companies that this was viable way to develop a medicine, this was this talking point. They were one and done. Certainly in the retina, the first one, that's definitely the case. But now as we've expanded to other areas like diabetes or like renal disorders, where we may need to provide a sustained effect over a longer lifespan, it's certainly not ever going to be frequent. The durability is between 5 and 10 years in most tissue compartments that are not like the brain or the heart. So, it's maybe 2 or 3 exposures in the lifetime, which is not bad, right?
That's still
And one of the
fantastic.
ways that companies like the one that developed a hemophilia treatment is considering that if an insurance company pays for the therapy, they wouldn't pay for it again. That treatment would be their lifetime cost. So it really puts the burden on the drug developer to figure out how to sustain that effect, because it's that the business model is changing. Traditional pharmaceutical development always relied on taking something again and again and again. So this was very against the traditional model.
But it also gives an incentive to the pharmaceutical company to develop it if they have to treat more than once. Are there more questions here? So we have a few minutes. You have also worked on Pompe disease, but you never mentioned anything. Would you just like to briefly tell us?
Yeah, the Pompe story is a little like SMA in its evolution. The original notion, the pathologist who described Pompe disease made that observation based on the heart abnormalities and on the skeletal muscle weakness. We've now learned over the past 10 years that this also is very much responsible. The weakness is due to the glycogen accumulation in the brain. So this has added to the complexity, but we have done several gene therapy studies in Pompe patients, not yet with the systemic treatment, just the local regional therapies initially. And in fact, in Pompe patients, we're the first ability to show that we can give the AAV vectors more than once. The next step in that journey is to utilize some of the new tricks to get more of the product in the brain. And then we'll continue with the effort to treat infants with Pompe and not just the adults. So the other Pompe studies that are out, we're the only ones who've done studies in the infant population, but we'd like to continue that because it's the greatest magnitude of effect. Normally those babies don't survive past 8 months of age. And we know that, at least in the models that we have, that they can be effectively cured, just like in SMA.
Thank you. Are there any other questions?
The genetic disorder called left ventricular non-compactable
Mhm.
cardiomyopathy. Is that one of them that you're?
Well, one form of it is called Bar syndrome, and we have studied Bar syndrome. Foundation was actually founded here in Florida by a mom who had two affected boys, another X-linked disease, actually. So we have a program to treat. There are only 50 patients in the U.S., so talk about rare. But all know one another. They're like brothers, and that's a goal, because we think it's very easy to measure the outcome in Bar syndrome, both heart function and skeletal muscle function. There's one more question. Possible to gene edit parents carrying these traits? Seemingly possible, but the focus has really been on the affected individuals, not the carriers. And now that the genetic testing is so readily available at a much reduced cost of how many people have ever participated in 23andMe or any of those things? Relatively inexpensive, right? In the beginning, to do whole genome sequencing or exome sequencing was about $2 million, and now it's $50. So, it's quite a revolution to get that genetic information and determine susceptibility. So that's where the focus is, rather than editing germ cells, which will probably never be ethically allowed, right? To avoid passing on the trait, one would have to edit the germ cells and not other cells in the body. So that is too much of an ethical dilemma. I think no one would want to see that, because it opens the door to a whole variety of things. But certainly carrier status and identifying individuals at risk, like the gene panels that we can do now just by cheek swab or spit testing and mail it back, and 2 weeks later, we often find the diagnostic dilemma faced by some patients is now being solved by doing genetic testing of all the genes all at once. And then particularly helpful in children who have a not recognizable condition. If you sequence the child and both parents, this helps sort out very quickly what the real problem is. So yeah, a lot of that opportunity exists because there's testing.
Thank you. Thank you very much, Barry, for coming out. This was fabulous, very impressive.
Thank you.
So next week we have Barry Carter, and I believe he's speaking on hemophilia. Yeah, so see you then. Thank you. I'm sorry.